Coming to the cellular organelles. We are not going to see all the cellular organelles. Few important cellular organelles and its some functions. Okay. So first one is the endoplasmic reticulum. Where is the endoplasmic reticulum present? Close to the nucleus. Okay. In the endoplasmic reticulum, we have two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. What is the major role of rough endoplasmic reticulum? It is involved in the production of ribosomes. Ribosomes present in the surface of the endoplasmic reticulum, giving the rough surface. So the name rough endoplasmic reticulum, which is having a major role in the production of proteins. Okay, so this is having the major role in the production of proteins. And smooth endoplasmic reticulum, how the surface will be? How the surface of the smooth endoplasmic reticulum? Very smooth. Is there any substances produced by the smooth endoplasmic reticulum? Yes, it is also involved in the production of few things like steroids. And it is also having the role in the detoxification mechanism. Which organ is involved in the detoxification? Liver. So this smooth endoplasmic reticulum, since it is having the major role in the steroid synthesis and in the detoxification process, which organ will have excess of smooth endoplasmic reticulum? Adrenals. Adrenal gland, which is the one producing the steroid hormones, the catecholamines, cortisols, and estrogen, other things through the catecholamine pathway, and the liver, since it is involved in the detoxification mechanism. These two organs have excess of smooth endoplasmic reticulum in its cellular organelles. Okay, and now this is about the endoplasmic reticulum, and we will also see about Nissl granules or Nissl bodies. Where you come across this Nissl bodies usually? Nissl granules, Nissl bodies. What is this? Yes, where is the Nissl granules? Near the, what is this part? What is this entire structure? Neuron. Everyone is aware of this? Yes, this is neuron and this is axon hillock. And where is the Nissl granules? They are the rough endoplasmic reticulum of the neurons. So this Nissl granules is the one which is having a major role in the synthesis of which chemical in the neuron? What you term it as? Neurotransmitters. Okay. So the main role of rough endoplasmic reticulum is in the synthesis of proteins, enzymes, because of the ribosomes. This is modified in the neurons as Nissl granules, which is the rough endoplasmic reticulum of the neurons, which is having a major role in the synthesis of neurotransmitters. They are the chemicals which is giving the signaling mechanism within the neurons. Okay, so that is about the rough endoplasmic reticulum. And there are also endoplasmic reticulum which has free ribosomes. This free ribosomes, it remain unattached with any membrane. It will not get attached to the any of the membrane. So it remain unattached to any membrane. Okay, and that is the site of synthesis of cytosolic and organella proteins. So that is called as free ribosomes. They do not get attached with any of the membrane. It remain unattached. So that is the site of synthesis of cytosolic and organella proteins. Okay, and which is rich with this free ribosomes is where you will see is the mucus secreting. Where is the excess secretion of mucus? Any idea? Goblet cells. Where is the goblet cells? In the small intestine. Okay, so the goblet cells which is secreting the mucus, which is the internal covering, inside covering of the small intestine. Okay, so that is one of the example of free ribosomes. The rough endoplasmic reticulum, which is having the free ribosomes, it means they are the ribosomes which is not attached to any of the membrane, and its one example is the mucus secreting goblet cells in the small intestine. Okay, here. So that is what is about the endoplasmic reticulum. Clear with the endoplasmic reticulum? Rough and smooth endoplasmic reticulum? Yes. And the endoplasmic reticulum in the neuron is micelle granules, and in the small intestine it is goblet cells which is having the free ribosomes. Okay, clear? And now we will move on to the Golgi apparatus. So what is the role of endoplasmic reticulum in general? Whether it is rough or smooth, synthesis of chemicals in the form of proteins, steroid hormones, or detoxification mechanism by some chemicals is going to happen. Okay, so overall it is producing, it is secreting the substances. Those substances has to get packed in a cover, or else it will freely float in the cellular substances inside the cytoplasm of the cell. So that packing mechanism is mediated by this Golgi apparatus. So whatever substance, wherever the location, whether it is in small intestine or the neuron or liver or wherever it is, okay, whatever substances is produced by the rough endoplasmic reticulum, that chemical substances will be packed inside the pouch-like substance. That structure is given by this Golgi apparatus. So here you will have the endoplasmic reticulum, rough endoplasmic reticulum, and it is releasing the proteins, ribosomes. So those ribosomes will be packed by the structure called as Golgi apparatus. So the main function of Golgi apparatus is it is giving the packaging of the substances. When you see about the structure of the Golgi apparatus, you will have the cisternae. So these are the cisternae, the foldings which is present in the Golgi apparatus. They are called as the cisternae. And you have two surface, cis phase and the trans phase. This cis phase is facing towards the endoplasmic reticulum. Whether it is rough or smooth, the cis phase of the Golgi apparatus is towards the endoplasmic reticulum, and the trans phase of the Golgi apparatus is towards the plasma membrane. Okay, why these two contents you have to consider is based on this cis phase and trans phase, we are going to see the function of Golgi apparatus. Okay, so what is the main function of the Golgi apparatus is packaging of chemicals as vesicles. Okay, so whatever is being newly synthesized by the endoplasmic reticulum, it is packed by the surface. The pouch-like structure is given by this Golgi apparatus. So these are all the incoming transport vesicles. So this one, all see here. you can see the enlargement. It is forming the bubbling of the structures, and on that contains the chemicals, and then it gets detached from the entire Golgi apparatus, and now it will freely float in the cytoplasm as the vesicle which is containing the chemicals. Okay, so that is the main function. And another important function of the Golgi apparatus is in the post-translational modification of proteins. Where you come across the term post-translational modification? Replication, transcription, translation. Replication is, what is replication? Dividing. And transcription, DNA converted to mRNA. Once when it is transformed to mRNA, that is the one which is giving rise to the proteins. So there you have to do with the post-translational modification of the proteins to give rise to the structure of various proteins. So that post-translational modification of proteins is happening because of the Golgi apparatus. And this is mainly because of the specific thing called as N-linked glycosylation that begins in the rough endoplasmic reticulum. So rough endoplasmic reticulum is the one which is giving rise to the proteins. So from the rough endoplasmic reticulum, the N-linked glycosylation, okay, which is beginning in the rough endoplasmic reticulum, and then other one is O-linked glycosylation. Then sulfation, phosphorylation. So these are all the functions which is mainly mediated in the post-translational modification of the proteins. Okay, and then next, this post-translational modification of the proteins, it is also followed with the another mechanism that is called as protein sorting and packaging. So what is happening in this protein sorting and packaging is, this protein sorting, it will sort out various proteins based upon its function. Some example is insulin. They are packed in the vesicles called as clathrin coated vesicles, and other cell membrane proteins. Hormone receptors. Example for this is hormone receptors. They are packed in, this is packed in the clathrin coated proteins. This is packed in non-clathrin coated vesicles. Okay, this clathrin is when you have the involution of the cell membrane, you have the receptors like this. Okay, so when this protein content is coming, so some will have the clathrin coated and some will be present without the clathrin coated. Okay, so insulin is mainly packed in the vesicles which is having the clathrin proteins. They are clathrin coated vesicles. And other cellular membrane proteins like hormones and the hormone receptors, all these are packed into the vesicles which is non-clathrin coated vesicles. Just remember what is clathrin coated vesicle, non-clathrin coated vesicles, and examples. Okay, no need to go in detail of all those things. So this is what is about the Golgi apparatus. And in the Golgi apparatus, we have seen the cis phase and the trans phase. Why we have seen those two phases is because of the COP proteins. They are the coated proteins in the Golgi apparatus, which is going to involve in the transmission of the proteins which is produced in the rough endoplasmic reticulum. So this is the cis phase and this is the trans phase of the Golgi apparatus. I have said the cis phase is the one which is facing the endoplasmic reticulum, and trans phase is the one which is facing the plasma membrane. So you have the COP proteins. You have two types of COP proteins, COP1 and COP2. Okay, and along with this, you will also study about the clathrin. So why we are dealing with this COP1, COP2 and clathrin is those substances which are produced in this endoplasmic reticulum is being transferred to the Golgi apparatus by the protein called as COP2. So this is involved in anterograde transfer. Okay, and what is the role of Golgi apparatus? To pack the chemicals which is produced. During the machinery process, during the manufacturing process, there is chances of producing improperly structured or organized substances. If those improperly structured substances is packed into the vesicle, then it will result in an improper function, right? So this will undergo a quality checking process. So during that packaging in the Golgi apparatus, if it is not being produced properly, improperly produced substances by the ribosomes, by the endoplasmic reticulum, will go back to the endoplasmic reticulum for the reorganization. So that is happening by this COP1 proteins. This is called as retrograde transmission. So you have two types of COP proteins, COP1 and COP2. COP2 is mainly involved in the anterograde transmission from the endoplasmic reticulum to the Golgi apparatus. And COP2 is retrograde transmission from the Golgi apparatus to the endoplasmic reticulum. Okay. And then third one, something you have to transfer from the Golgi apparatus. After it has been packed into the vesicles, it has to move towards the plasma membrane. The vesicle will get ruptured with the plasma membrane to release the neurotransmitter or to release the hormones in the target site. Whatever may be the thing packed in the vesicle, it should have some action in some part of the body. So the vesicles will move towards the plasma membrane, and then that is being transferred to the plasma membrane by this clathrin proteins. Okay, so this is how the transmission is taking place. Molecular level, all these needs the stimulus, signal, action potential, everything. But the general mechanism is, this is how it is transferring. If there is any defect in the COP proteins, the substances produced by the endoplasmic reticulum will not go to the Golgi apparatus for packaging. If there is any defect in COP1, you will get the result as improperly produced substances, and that will result in improper function. You will come across various clinical conditions. These are all the improper things happening in the mutation cases. Most of this kind of abnormalities you will come across as a genetic condition, mutated condition. Okay, here. So this is what is COP proteins. And next one is the peroxisomes. What is the main role of peroxisome? Any idea? The peroxisomes is having a major role in the beta oxidation process. The beta oxidation of the fatty acids. It is mainly involved in the catabolism of lipids, especially very long chain fatty acids. And this peroxisomes, as the name, it is involved in the oxidation process. It results in the production of hydrogen peroxide. So which is having the major role in the oxidation process. Okay, so that is about the peroxisomes. This is the structure, one of the organelle which is present in the cell, which is mainly having the catabolic role of various lipid substances, beta oxidation of fatty acids, and especially the catabolism of very long chain fatty acids, and it is having the major role in the oxidative mechanism, okay, in the production of hydrogen peroxide. What is meant by oxidation and antioxidant? You have any idea about oxidation and antioxidant production? What is happening in the oxidation process? Which is good? Production of oxidants or antioxidants? What is oxidants? See, for example, just a small thing I will tell you. This is the cell which is having the electron, and now you have a defective electron in this cell. So this defective electron you will consider as free radical. Okay, so this is now you call as oxidant. Production of free radicals. Okay, what this oxidants will do is it is now having one oxygen as a defective thing. So its aim always it will try. See, for example, I am writing and I am going to draw. I have only pencil, I don't have eraser. If I draw by mistake anything wrong, I need eraser to erase, but I don't have eraser. But my friend neighbor is having the eraser. There is a rule, you should not get things from your neighbors. But I have drawn something wrong. Now I need the eraser. What will be my aim? Always how to steal the eraser without anybody's knowledge. So that will be the aim of my thought. So as like this, here there is a deficiency of oxygen, and its aim is to get the oxygen and to make the deficiency as normal. So where it will go? It will go to the normal cell which is having complete oxygen. So immediately what will happen? It will steal one oxygen and make this as a defective cell. And this now will become oxidant and it will steal from the another cell. This is how the free radical oxidative damage is taking place in our body. If there is no production of antioxidants, what is the antioxidants? It is giving the extra oxygen to the oxidant. So this cell is now having the defective oxygen. The antioxidants is like someone is coming and donating the eraser in the classroom free of cost. So now I am sufficient. Oh, I got the eraser. Even if I draw wrong, I can erase. No need to steal from the neighbor. That thought is gone because I got the eraser. Okay, so that is how the antioxidants, it is also produced in our body. If the mechanism is normal, the antioxidants will be normally produced by our own body cells, which will donate the free oxygen and make this stealing process not to happen. So the adjacent production of free radicals will not take place. Okay, so this is what is called as the oxidative damage. Okay, so this oxidative damage, all these oxidation and the free radical production, oxidants, everything is been happening in the peroxisomes. Okay, clear? And now coming to the mechanism called as lipofuscin. Have you come across the term lipofuscin? Yes? What are all the things you are seeing here? Here? Any idea about lipofuscin? Yes? It is the mechanism of lipochromes. What is this lipochromes? They are the lipid containing residue of lysosomal digestion. The lysosomes as like vesicles, it is packed inside the cellular organelles which is having rich of enzymes. Those enzymes is involved in the digestive mechanism of the own cells. If the lysosomes is getting ruptured, our own cell will be damaged by mistake or by any abnormal condition, lysosomal rupture inside the cell will digest our own cell. Okay, so this lysosomal damage, if this digestion process, which is rich with the lipid containing residue, those lipid containing residue will get accumulated. The most common locations where the accumulations we will find is eyes. In the eye, we will see this lysosomal digestion residues. And this is also seen in the heart. This is how the normal heart you see. And here you see the yellowish discoloration in the gross structure. Okay, but most commonly we will not see the gross for our examination purposes. We will see the histological picture. So you see the brownish deposition. Okay, that is one of the indication of lysosomal digestion, lipid containing residue. And you will also come across this in the yellowish discoloration in the eyes, not in the conjunctiva. If you see the yellowish discoloration in the conjunctiva, that is jaundice. But here you will see in the inner ball of the eyes. Okay, so that is just thing about the lipofuscin. And next, about the cilia. Where you will have the cilia? What is cilia? What is flagella? What is cilia? Motile structure. Okay. Mostly this cilia is present in where? Inside of the cell or outside of the cell? Outside of the cell. See, you have the cell. You have the flagella, the long motile structure, and you have a small cilia-like structures. Which organ is having the cilia mostly? Endometrium of the uterus, small intestine, and the respiratory system. Okay, so which will expel the irritant substance by cough mechanism and sneeze mechanism. Because of the ciliary bead, we are getting the cough reflex and the sneeze reflex. So these are the structures which is abundant with the cilia, and these cilia have a major role in these structures. In the uterus, what is the role of cilia? It is having the major role in the movement of female reproductive system. Okay, the egg which is produced from the ovary, once when it is going to the fallopian tube, from the all over structures of the fallopian tube, see, you have the female uterus like this. Okay, so whatever egg is produced in the uterus, it has to travel all its way, and then once when it is fertilized, it will get implanted into the uterus, and the sperm which is traveling, the movement is given by the tail of the sperm as well as the ciliary mechanisms. Okay, so this cilia, it is just a small structure, but if there is any defect or mutation in the ciliary production, it results in infertility. Yes or no? If there is no movement of the egg, no movement of the sperm, it results in a clinical condition we will see as infertile. It is not only because of the absence of hormones, not only because of the decrease of sperm count, it is also because of a very small structure, cilia. Okay, and in the respiratory system, what you will have? The cilia, which is expelling the mucus, which will expel through the sneeze and cough reflex. Yes or no? The cilia which is present over here, the ciliary bead, either it will send it inside the lungs or it will expel it out, the irritant substances, as well as the mucus production. The mucus production, if it is not being expelled out, if it is gets lodged in the inside of the lungs, you will result in bronchiectasis, chronic sinusitis, repeated respiratory infections. If the ciliary bead is not working properly, all the irritants will get lodged inside the lungs. It will not do the function of sneezing and coughing reflex. So what happens? The irritants or the bacterial infections, viral infection, whatever may be the infectious substance, it will go inside the lungs and causes repeated lung infections. So this is what is about the cilia. When you see about the structure of the cilia, okay, microscopic view, macromolecular view, you will have the axonems. So these are the axonems. You have 9 doublets of axonems. So this is the cross-sectional view. The cross-sectional view, you have the 9 doublet faces of axonems. In the central, you have 2 axonems. So totally 9 plus 2. Okay. So this is being interconnected by the structure called as nexin. Just remember these 2 things, nexin and dynein. So these dynein arms are present in the outer surface of the axonem, and each axonem, the doublet faces, is connected by the structure called as nexin. Okay, so just remember these 2 things in the structure and the functions of cilia. So if there is any defect in the ciliary mechanism, it will come across with mainly the 3 systems, the lungs and the second one is reproductive system, infertility and chronic lung infections, repeated lung infections. And you will also have this role in the small intestine, the absorptive mechanisms, the movement of the bolus, the movement of the substances inside the small intestine for mixing and propagation of the peristaltic movements. Okay, so these are the 3 major clinical conditions which causes ciliary defect, and that is called as have you come across this syndrome anywhere? Cottagnous syndrome. This syndrome is having a triad of 3 conditions. What are the 3 triad of conditions? Yes, repeated lung infection, bronchiectasis, recurrent sinusitis, and third one is anyone? Situs inversus. What is meant by situs inversus? You know bronchiectasis, sinusitis. What is situs inversus? It is the one in the, what is this structure? What is this structure? And what is this? This is right side or left side? Left side. In situs inversus, the structures of the thoracic cavity is sitting in an inverse manner, like a mirror image. So the right side and the left side is inversed. So it is sitting in an inverse manner. If you see the mirror image, the right and the left side of the heart is located inversely. Okay, so that is what you will encounter in this Cottagnous syndrome. It is due to the mutation of the gene which is having a role in the ciliary structural maintenance. It is also a rare genetic disorder. Okay, clear? So that is what is about